Hello everyone, thanks Edo for introducing me. Thanks to the organizers. So in this slot I'm actually representing Roche, and I'm going to talk about the link between microbiome and immunity. So just a short introduction. So I'm actually a PhD student in University of Trento in Italy in the Nicola Segata's group, but last year I had the opportunity to spend 6 months as an intern at Roche in Basel, Switzerland. You can see the picture of the campus. The towers are actually the highest buildings of Switzerland. Then in the summer in the river people swim. So I had a great time. But so I was part of the research department in the microbiome team under supervision of Petar Stepanovic. And in the Roche research department, it's very big. Of course, many projects are focused on the drug development, but some of the projects, such as this one that I'm going to talk about, are very almost basic research, about disease understanding and so on. And then Roche even publishes papers, and so that's why I'm also able to talk about it here. To introduce the disease, so we are focusing on antiphospholipid syndrome, which is autoimmune disease, which symptoms basically it leads to blood clots, thrombosis, which can lead to stroke, miscarriage, and so on. It's relatively prevalent. The diagnosis is reduced to basically to testing of the presence of antibodies and also the symptoms. And since the cause is unknown, the medication is with the blood thinners to prevent the blood clots. And so there is a hypothesis, since, as you know, microbiome, there's so many microbes produce so many proteins, it produces a high antigenic load that the human immunity reacts to. So can there be some sort of cross-reactivity, that causes the autoimmune disease to develop? So to go into slightly more details, so the antibodies that can attack usually attack the phospholipids, or even more commonly the beta-2 GPI, which is a protein associated with phospholipids. And there are several actually antibodies, and the patients can be positive for either of them or even all three of them. And so the background work to this project comes from a group of Martin Kriegel, at that time at Yale. And they published it in a paper that you can easily find, in which they picked an example, this Roseburia intestinalis, which is a very common human gut bacteria. And they show that this protein, the DNA methyltransferase, has an epitope, which you can see in the panels A and B, that basically is homologous to the one of the beta-2 GPI. And they hypothesize that the epitopes targeting this Roseburia intestinalis DNMT could cross-react with the human beta-2 GPI and then lead to autoimmunity. So they took this protein and they confirmed in vitro, actually with immunoassays, that indeed this binding can occur. And they also proved in vivo in immunized mice that it can lead to disease-like symptoms. And moreover, they recruited a cohort of these APS patients and also healthy controls. And they checked their microbiome with 16S sequencing. They didn't find any differences in the microbiome composition, and found that this Roseburia intestinalis is quite prevalent in both groups. But thanks to collaboration of this Martin Kriegel's group and Roche took these metagenomic samples of even more subjects recruited and analyzed with metagenomics sequencing. Right, and then so that's what we did. So in total we have 34 subjects. Their samples sequenced to quite high depth and nearly 100 million reads on in median. So then we run the standard metagenomic analysis. We use metaflant4 for the species composition, and we confirmed, there's no difference in alpha and beta diversity, except some patients that were taking protein pump inhibitors that we had to exclude downstream or add as a covariate. But it's a detail. We also checked again this Roseburia intestinalis, and we indeed confirmed it's highly prevalent in both groups, which is also in line with the French reference healthy cohort. The relative abundance is comparable in the groups. And looking in the strain level, you can see on the right in the phylogenetic tree that the patients are mixed with the controls, with the healthy controls. So there's no, let's say, evolutionary strain level signal there. So we moved on to, let's say, another type of analysis, which is actually searching for the epitopes. As in the original publication, they were focusing just on the DNMT from the Roseburia intestinalis. But we thought, what other proteins could, potentially present this epitope for the cross reaction, and so on. So as a first step, we focused on the domain 1 of the beta-2 GPI, which is this RGG-MRK conserved epitope sequence, and we used a database that we have in the University of Trento, which with over 1 million of mags and their proteins in them. And also alternatively, we searched for this epitope sequence directly in the sequence reads. So in terms of a search in the database, so we were looking for the UNRF90 level protein families, and we confirmed these bolded ones are actually those DNA methyltransferase that was identified already in the original publication. But we saw that there are many more proteins that are prevalent, let's say, in the MUGs, which is the hits column, and many of them are not even in UniProt. They are, discovered, de novo in the MUGs, and some of them we could profile with human 3.8, some not. But for those we didn't find a significant difference among the groups. And so for the second type of analysis that we did for the epitope, we were checking basically this, epitope load or antigenic load in the reads, in the sequence reads directly. So we filtered, we were looking for the RGG-MRK in the reads, and by mapping to this, IGC catalog, making sure, the reads are in the correct, the RGG-MRK is found in the correct frame. And then we counted those hits and compared again between the groups, and we again didn't find, as you see in the box plots, we didn't find any difference, let's say, in this presence of epitope in the intermicrobial reads. So just to summarize, there are hypotheses that based on which there could be a link between microbiome and autoimmune diseases, in particular APS, via this cross reaction. And so we performed the first metagenomics analysis for this disease on the previous cohort. It was analyzed with 16S previously. We confirmed the 16S findings that actually there is no difference in the microbial composition between the groups. And also by more focused analysis on the epitopes, we didn't find the difference either. So this brings us to, let's say, some sort of speculation. So it means that it seems that the microbiome of both the diseased and the healthy people is kind of the same. These antigenic proteins and the species carrying those proteins are very prevalent in both of the groups. So it must depend on some other still on some other unknown factors on why this disease develops, and so basically further research is needed. Although these results are still preliminary, that they need to be expanded. So to be continued. So by this last slide, I would like to thank to everyone that participated on this project. Above all, I would like to thank to Petar, who was for this opportunity to do the internship in his team and for this project and everything. And I thank you, and I'm looking forward to the questions.